can we on to the next topic?
I'll present next, sir. My topic is physiology of pain. Definition: according to International Association for the Study of Pain, the pain is defined as an unpleasant sensory and emotional experience associated with actual or potential tissue damage or described in terms of such damage. Thus, pain has objective physiologic sensory aspects as well as subjective emotional and psychological components. The term nociception refers to neural response to traumatic or noxious stimuli or potentially tissue damaging stimuli. The processes involved in pain are transduction, transmission, modulation, and perception. Transduction refers to where the noxious stimuli is converted into electrical signals in the sensory nerve endings. In transmission, the conduction of electrical signals to the central nervous system occur. Modulation, either inhibition or facilitation of pain is done. Transduction. Nociceptor is the primary sensory structure for transduction. Most nociceptors are free nerve endings that sense heat, mechanical and chemical tissue damage. The mechanoceptors respond to pin, prick, and pinch. The silent nociceptors, they respond only in the presence of inflammation, whereas polymodal mechanoheat nociceptors, they respond to excessive pressure, excessive temperature, and extremes of temperatures like more than 42 degrees Celsius and less than 18 degrees Celsius, and algogens and pain-producing substances like substance B, calcitonin, G-related peptides, prostaglandins, thromboxanes, leukotrienes, etc. The vanilloid receptor 1 is isolated from the sensory neurons. These vanillins include capsaicins that cause pains. The VR1 receptors responds to pain, protons, and temperatures more than 43 degrees Celsius. The another receptors are VRL1 receptors, which respond to temperature more than 50 degrees Celsius, but not to capsaicins. They are isolated from the C fibers. Transmission. Pain impulses are transmitted by two fiber systems. One is unmyelinated C fibers and another one is myelinated A delta fibers. The unmyelinated C fibers are 0.4 to 1.2 micrometer in diameter with a conduction speed of 0.5 to 2 meter per second. They carry slow, dull, and poorly localized pain. Myelinated A delta fibers are 2 to 5 micrometer in diameter with a conduction speed of 12 to 30 meter per second. They carry fast, sharp, and well localized pain. The dorsal horn of the spinal cord consists of 6 laminae. The unmyelinated C fibers synapse and terminate in the lamina 1 and 2, whereas A delta fibers terminate in lamina 1 and 5. The synaptic junctions between the first order neurons and the dorsal horn cells are the slides of plasticity. The dorsal horn cells are called as a gate, where the pain impulses can be gated or modified. The gate control theory. This gate control theory was first proposed by Ronald Melzack and Patrick Wall in 1965. According to this theory, the painful information is projected to the supraspinal regions if the gate is open, and pain is not felt if the gate is closed by simultaneous inhibitory impulses. Like A beta fiber, which has mechanoceptors, which activates the inhibitory interneurons and close the gate and inhibits the painful stimuli. Examples of these are massage, acupuncture, and transcutaneous electrical nerve stimulation. A delta and C fibers are nociceptors. They inhibit the inhibitory interneurons and open the gate and transmits the painful stimuli. The second order neurons consist of wide dynamic range neurons and nociceptive specific neurons. The wide dynamic range neurons respond to nociceptive and non-nociceptive stimuli from A delta, C fibers, and A beta fibers. They are most abundant in lamina 5. The nociceptive specific neurons respond only to high threshold nociceptive stimuli. They are arranged somatotopically in the lamina 1. The axons of the second order neurons cross the midline on the anterior commissure of the spinal cord to ascend as the spinothalamic tract and ends in the thalamus, reticular formation, nucleus raffimagnus, and periaqueductal gray matter in the midbrain. The spinothalamic tract can be of two types, which is lateral neospinothalamic tract or medial paliospinothalamic tract. The lateral spinothalamic tract ends in the ventroposterior lateral central nucleus of the thalamus and is responsible for the location, intensity, and duration of pain. Whereas the medial or paleo-spinothalamic tract projects to the medial thalamus, which are responsible for the autonomic or emotional aspects of pain. The perception is the subjective feeling of the painful stimuli. The third order neurons in the thalamus project to the somatosensory cortex 1 and 2 in the postensorial gyrus and superior wall of Sylvian fissure, respectively. This perception of the discrete localization of pain takes place in these cortical areas. The other projection neurons engage the cingulate cortex, insula, and amygdala via the parabrachial nucleus. These structures mediate the suffering and emotional components of pain. Modulation occurs via peripheral and central structures. Peripheral modulation occurs in nociceptors and in the spinal cord, either inhibit or facilitate the pain, whereas the central modulation occurs in the supraspinal structures. Peripheral modulation. The nociceptors display sensitization following repeated stimulation that results in decrease in threshold, increase in frequency response, decrease in latency of the response, and spontaneous firing even after cessation of the stimulus. They can cause hyperalgesia. Hyperalgesia is the enhanced response to the given noxious stimuli. The hyperalgesia can be primary hyperalgesia and secondary hyperalgesia. The primary hyperalgesia occurs at the site of injury due to interaction between the pro-inflammatory mediators and their receptors like histamine, bradykinin, prostaglandin, and leukotrienes. Whereas the secondary hyperalgesia occurs in the surrounding tissues where it is manifested as triple response, whereas flare, local edema, and sensitization to the noxious stimuli. It is due to neurogenic inflammation and due to antidromic release of a substance P from the collateral axons of the primary afferent neurons. Central modulation. The central modulation either facilitates or inhibits pain. The facilitation mechanisms include wind up and sensitization of the second order neurons, receptive field expansion, and hyperexcitability of the flexion responses. The neurochemical mediators like substance P, calcitonin G-related peptide, VIP, cholecystokinin, angiotensin, glutamate, and aspartate. These changes trigger changes in the membranal excitability and mediates via G protein coupled receptors, which stimulates the intracellular second messengers and increase and phosphorylate the substance to increase the intracellular calcium. The excitatory transmitters like aspartate and glutamate also stimulate NMDA receptors and increase the intracellular calcium. These stimulates the nitric oxide synthase, which synthesize the nitric oxide. The nitric oxide diffuses out of the neurons, and by action on the guanyl cyclase, they stimulates a cyclic GMP formation in the neighboring neurons, which can either excitatory or inhibitory. Then inhibitory mechanisms. Inhibitory mechanisms can be segmental or supraspinal. The segmental mechanisms consist of activation of large afferent fibers, mediating inhibition of the wide dynamic range neurons and the spinothalamic activity. The inhibitory neurotransmitters include glycine and GABA. They are mediated by GABA B receptors, which increase the potassium conductance across the cell membrane. The supraspinal inhibition. The supraspinal inhibition occurs whereby several supraspinal structures send fibers down the spinal cord to inhibit the pain at the level of the dorsal horn. These include periaqueductal gray matter, reticular formation, nucleus raphe magnus. They act presynaptically in the primary afferent neurons and postsynaptically in the second order neurons or the interneurons. They utilize monoamines like noradrenaline or serotonin as neurotransmitters and transmit the nociceptive stimuli in the spinal cord as well as on the spinal inhibitory neurons, which store and release the endogenous opioids. The endogenous opioids are mediated via enkephalins and beta endorphins. They act presynaptically, whereas the exogenous opioids acts on the postsynaptic second order neurons. Physiological responses to pain. The somatic and visceral pain fibers are fully integrated with the skeletal motor system and sympathetic system in the spinal cord, brainstem, and the higher centers. These synapses are responsible for the reflex muscle activity associated with pain. The reflex sympathetic activation occurs and causes catecholamine release locally and from the adrenal medulla, which increases the heart rate and increases the BP and increases the myocardial work and metabolic rate and increases the oxygen consumption. In gastrointestinal system, the gastrointestinal tone is reduced and which delays the gastric emptying, may cause paralytic ileus, and in urinary system also the sphincter tone is increased, causing urinary retention. Hormonal response, increases secretion of catabolic hormones and decreases secretion of anabolic hormones occur. Metabolic response include hyperglycemia due to gluconeogenesis and decrease in insulin secretion, increased protein metabolism and lipolysis occur. In respiratory system, hyperventilation due to stimulation of the respiratory center and hyperventilation due to splinting and muscle spasm in the abdomen and thorax surgeries. Diencephalic and cortical responses include anxiety and fear. Psychological mechanisms includes deleterious emotional effects. So it's
Okay. So all of you should have read about the pain pathway from your physiology days. And, as anesthesiologists, we do have to have a very clear concept about how pain is transmitted in the human being, and that will help you to manage pain better. If you know the pathway, what are all the interventions that you can do at various levels. And that is the reason why this question is repeatedly asked in your exams, so that you will have an idea about how to manage an acute pain and a chronic pain. Okay, so that is the reason why that has been asked, and we have dealt with that very nicely.
thank you, sir.
Just I will share my screen with you.